Hello again. Today we're going to be reading another article by Dr. Ray Peat. You can find this on his website, raypeat.com, under the articles section. And this one is part one in a two-part series, and it's called The Problem of Alzheimer's Disease as a Clue to Immortality. And this is part one. And again, this is for educational purposes only. Hopefully this will help you in your research, and I'm sure you will learn a lot through this article. Please like and subscribe. Share this wherever you're on social media to get the word out, and I would greatly appreciate it. So, this is a lot of information, so it's broken up into two parts, and you kind of have an outline of how it will flow. You can see we're starting with the introduction. Okay, this is how it goes. The toxicity of estrogen and of the unsaturated fats has been known for most of the 20th century, and much has been learned about their interactions in the aging process. The body during this time has been understood as a dynamic interaction of cellular trophic influences which govern both form and function. My argument here will be that some of our adaptive, protective regulatory processes are overridden by the excessive supply of unsaturated fats, supported by a few other toxins, in our diet, acting as a false signal system, and that cholesterol, pregnenolone, and progesterone, which are our main long-range defenses, are overcome by the effects of the unsaturated fats, and that the resulting cascade of ineffective and defective reactions, including various estrogen-stimulated processes, leads to lower and lower energy production, reduced function, and death. At certain times, especially in childhood and old age, iron, which also has important regulatory roles, accumulates to the point that its signal functions may be inappropriate. It interacts with estrogen and unsaturated fats in ways that can change restraint and adaptation into sudden self-destruction, apoptotic cell death. If we look at the human organism from one perspective, it seems coherent and intelligible. But from the perspective of established academic biological doctrine, it seems appallingly complex, lacking any visible integrating principle, and as a result, simplistic mechanical, pharmaceutical, or religious ideas are increasingly offered to fill the gap. But experimental data can be taken out of the muddle and put to coherent human use. In what follows, I am acting as though the doctrines of genetic determination and regulation by membranes were more historical relics. The emerging control systems are now clear enough that we can begin to use them to reverse the degenerative diseases, Alzheimer's dementia, epileptic dementia, arthritis, osteoporosis, depression, hypertension, hardening of the heart and blood vessels, diabetes, and some types of tumor, immunodeficiencies, reflex problems, and special atrophic problems, including clearing of amyloid and mucoid deposits. I think many people experience regenerative age regressing when many circumstances are just right. For example, taking a trip to the mountains in the spring with friends can optimize several basic regulatory systems. Can optimize several basic regulatory systems. There we go. Okay, hopefully the wind over here is not too much. I don't have the best mic, and I'm sure it's picking up some of the wind from outside. So hopefully it's not too distracting. Okay. Part 2. Common factors in brain injury during growth and aging. Most people are surprised by the number of cells in the prenatal brain and in the very old brain. In the human fetus at 6 months of development, there are about twice as many brain cells as there are at the time of birth. And in old age, the number of cells in the brain keeps increasing with age. So that at the age of 90, the amount of DNA in the brain 36.94 grams, is about 50% greater than at the age of 16 to 20, 23.04 grams. In the aged brain, glial cells multiply when neurons die. In the fetus, the cells that die are apparently nerve cells that haven't yet matured. The factors that are known to reduce the brain size at birth are also factors that are involved in the degenerating brain in old age or Alzheimer's disease. Lack of oxygen, excess unsaturated fats or deficiency of saturated fats, estrogen excess, progesterone deficiency, and lack of glucose. A lack of carbon dioxide is probably harmful in both. Inflammation and blood clots may be factors in the aging brain, and bleeding with vascular spasm is sometimes a contributing factor to brain damage in both the old and the fetal brain. Endotoxemia may be a factor in nerve degeneration only during adult life, but it is sometimes present during pregnancy. And this is from MC Diamond, Enriching Heredity, the Importance of the Environment on the Anatomy of the Brain. All right, part 3. A view of entropy, renewal of the brain. When a fertilized egg is developing into a person, each cell division creates a new environment for the daughter cells, to which they adapt. They may run into limits and resistances. Sometimes a certain gene doesn't meet the need of the situation, or toxins are present, or nutrients and hormones are imperfectly supplied, but the process is flexible, and a way is normally found to get around the limitation. The embryo's brain development is my favorite example of the ways genes interact with the environment. We might think of the optimal brain development of a person or a rat or a chicken as something which is clearly limited by the genes. But if rats are given a stimulating environment, each generation gets a slightly bigger, slightly more intelligent brain. If rats are treated during pregnancy to increase the amount of progesterone, the offspring have bigger brains and learn more efficiently. Still, that might just be restoring a condition that was natural for rats in some perfect environment. Chickens develop inside an egg shell, and so the nutrients needed for their development are all present when the egg is laid. The brain, like the other organs, stops growing when the food supply is used up, but an experimenter, Zamenhof, opened the eggshells at the stage of development when the brain normally stops growing, and added glucose, and found that the brain continued growing, producing chickens with bigger brains. The genes of a chicken, as part of a system, have something to do with the development of that system, but the environment existing in and around the organism is able to guide and support the way the system develops. The size, complexity, and intelligence of the brain represents a very large part of the information contained in the organism, and Zamenhof's experiment showed that the ability to realize this potential to create this complexity comes from the support of the environment, and that the genetic nature of the chicken didn't constitute a limit to the development of its brain. I'm going to argue that Alzheimer's disease is analogous to the situation confronted by the developing chicken embryo or the rat or human fetus. When the environment is unable to meet the needs of the highly energetic, demanding, and sensitive brain cells, and the brain cells begin to die instead of developing into a more complex state, passing beyond various barriers and limitations. There are two stereotypes that are in conflict with this view. One, that the structure of the brain is determined at an early point in life, sometimes explicitly stated as the age of 12 or 16, and two, that the structure of the brain goes into an entropic deterioration during the process of aging. My position is that the brain cells are in a vital developmental process at all times, and that the same things that injure the brain of a fetus also injure the brain of an aging person. If novelty is really appearing during development, then it is hard to maintain that entropy increases during the development of an individual. Isn't a child a richer organization than a fertilized egg? Isn't an adult more individualized or realized than an infant? Seen from the inside, our known world gets richer with experience. Learning is certainly anti-entropic. Where does the idea of increasing entropy with living come from? Many things contribute, including a doctrine of genetic determinism, the old Platonic idea of the imperfection of the concrete, the unreality of the existent, and the medieval idea of the corruption of the body. These philosophies still motivate some people in aging research. The astrophysicist N.A. Koitrev, probably said that incorrectly, showed that the idea of an entropic cosmos derived simply from the assumptions of 19th century deism. God set the clockwork universe in motion and left it to run down. Early in this century, Raymond Pearl argued that the rate of living governed the lifespan, so that fast living meant a short-lived. He based his argument on cantaloupe seeds. The faster they grew, the sooner they died. This was because he didn't give them anything but water, so they had to live on their stored energy. If they grew quickly, obviously they ran out of stored energy sooner. I've never heard that described as a stupid idea, but I think politeness is sometimes carried too far. In the clock analogy or the seed analogy, the available energy is used up. The clock with its wound-up spring and the seed in a dish of water may be considered as closed systems, and we can understand their fate. But if it is foolish to argue from a confined seed to free-living organisms, then it is just as foolish to argue from a clock to a cosmos. Unfortunately, these inferences about closed systems are often applied to real situations that aren't energetically closed. The quote-unquote rate of living theory of aging picked up the idea of aging as a natural physical property of time and gave it expression in mathematical form, arguing that the total lifetime entropy production could be calculated to give insight into life expectancy and evolutional development. Unfortunately, the equation Hershey used assumed that the flow of heat out of the body into the surroundings is reversible. This suggests an image of Dr. Frankenstein vivifying his monster with lightning, putting the heat back into the body. If heat is to be put back into the body, it is necessary to make sure that it is appropriate for the structure as it exists. And in that paragraph, we're referencing Hershey in entropy, basal metabolism, and life expectancy. Okay, moving on. Actually, it is just the directed flow of energy which generates the structures. If any biological argument can be made from the idea of entropy, it is that it would be extremely difficult to regenerate food by putting heat into a person. In a few situations, it is possible to show that living structures can directly absorb heat from their environment, causing the temperature to fall, or negative heat production. But the exact meaning of this isn't clear. That's from B.C. Abbott et al., called The Positive and Negative Heat Production Associated with a Nerve Impulse. Okay, and there's several references in there. All right. It is now clear that in both crustacean and mammalian, this is from Howarth et al. 1968, non-myelinated fibers, there is an initial production of heat during or soon after the action potential, 80% of which is rapidly reabsorbed. A.I. Zotin, okay, here's a quote, aging and rejuvenation from the standpoint of the thermodynamics of irreversible processes. Sorry, I think that's the title of that document. Hard to follow exactly how this is supposed to flow, but all right. So then, citing the theory of Prigogine-Wyam, argued that the aging process involves both a decrease in entropy and a decrease in the rate of heat production. Okay. Hopefully you are following along visually as well, because that was a rough paragraph. All right, continuing on. Regeneration involves the production of entropy, as when an egg is formed. The temperature fluctuation at the time of ovulation might make a contribution to the construction of the entropic egg. The argument that aging of the animal, like aging of the cosmos, is governed by, quote unquote, the tendency of entropy to increase, has led people to say that rejuvenation would be like unscrambling an egg. Zoten's argument is interesting because he says that an egg is a scrambled animal. This view is very much like Warburg's and St. George's theory of cancer, that it is like a reversion to a simpler state of life. To sketch out what I have argued in different contexts, water is the part of the living substance that we can most meaningfully discuss in terms of entropy. In fact, much of the concept of entropy has derived from the study of water, as it changed state in steam engines, etc. Cancer cells, like egg cells, have a higher water content than the differentiated functioning cells of an adult, and the water is less rigidly ordered by the cellular molecules. This different, more mobile state of the water can be measured by the NMR, or nuclear magnetic resonance machines, which are used for MRIs, or magnetic resonance imaging. Estrogen has a special place in relation to the water in an organism. It is intimately involved with the formation of the egg cell, and wherever it operates, it increases both the quantity of water and apparently the disorder of the water. Its function, I believe, is to promote regeneration, as in Zoten's scheme, by increasing entropy or scrambling the animal. The way it promotes regeneration is by promoting water uptake, stimulating cell division, and erasing the differentiated state to one degree or another, providing a new supply of quote-unquote stem cells, or cells at the beginning of a certain sequence of differentiation. These more numerous cells then must find a hospitable environment in which to develop and adapt. If the proper support can't be found, then they will be recycled, like the unfed cells in the brain of a fetus. If we imagine the course of development as a summary of evolution, ontogeny recapitulating phylogeny, then the egg, as it quote-unquote unscrambles itself in embryonic development, passing through stages resembling jellyfish, worm, fish, reptile, bird, baboon, keeps finding that the available energy allows it to, in effect, say, I want this, I don't want that, until it emerges as a human baby saying, I want, and begins eating and learning, and with luck continues the unscrambling or self-actualization. Degenerative aging, rather than being physically derived from the properties of time, seems to be produced situationally by various types of contamination of our energy supply. Unsaturated fats interacting with an excess of iron and a deficiency of oxygen or usable energy redirect our developmental path. The saturated fats in themselves seem to have no signaling functions, and when they are naturally modified by our desaturating enzymes, the substances produced behave very differently from the plant-derived eicosanoids. As far as their effects have been observed, it seems that they are adaptive rather than disadaptive. All of the factors that affect the brain of a fetus should be examined in relation to the aging brain. Besides estrogen and fats, I am thinking of oxygen and carbon dioxide, glucose, iron and calcium, cholesterol, progesterone, pregnenolone, DHEA, the endorphins, GABA, thyroid, and vitamin A. An additional factor, endotoxic poisoning, eventually tends to intervene during stress and aging, exacerbating the trend begun under the influence of the other factors. Okay, part 4, false signals from the environment. The environment can be supportive, but it can also divert development from an optimal course. Passively taking whatever you are given by history and nature is entropic. Choosing intelligently from possible diets, selecting courses of action, will create pattern and reduce entropy. If education contains an element of choice and self-actualization, then the results seen in several Alzheimer studies could have a significance larger than what has been suggested by the investigators. A diagnostic bias has been reported to result from the use of standardized tests based on vocabulary, because education increases vocabulary and tends to cover up the loss of vocabulary that occurs in dementia. In the Framingham study, it was concluded that there was a real association of lower educational level with dementia, but the suggestion was made that self-destructive practices such as smoking were more common among the less educated. The Seattle study of the patients in a health maintenance organization showed a very distinct difference in educational level between the demented and the non-demented, both of whom had roughly similar frequency of prescriptions for estrogen. The features that seemed important to me that weren't discussed by the authors were that the demented women had a much lower rate of progestogen use and a much higher incidence of hysterectomy, which interferes with natural progesterone production. Although Brenner et al. In the Seattle study concluded that this study provides no evidence that estrogen replacement therapy has an effect on the risk of Alzheimer's disease in postmenopausal women, they reported that current estrogen use of both the oral and the vaginal routes had odds ratios below 1, while former use of both types yielded odds ratios above 1. They seem to neglect the fact that Alzheimer's type disease in old people has a long developmental history, so it is precisely the former use that is relevant. 31% of the demented women had formerly used estrogen, and only 20% of the control group. Since estrogen is a brain excitant, present use creates exactly the same sort of effect on verbal fluency as other signs of awareness of the environment that a little cocaine does. Anyone who neglects this effect is probably deliberately constructing a propaganda study. This observation that the demented had 155% as much former estrogen use as the normal group, as well as the difference in rates of progestogen use. Normal patients had 50% more progestogen use than demented, and hysterectomy. Demented had 44.1% versus 17% in the normals, i.e. 259% as many. The incidence of hysterectomies after the age of 55, which is a strong indication of a natural excess of estrogen, in the demented was 374% of the incidence in the non-demented. Okay, so let me reread, because that was a lot in parentheses, and I want to make sure that this thought is conveyed correctly. So this observation that the demented had 155% as much former estrogen use as the normal group, as well as the difference in rates of progestogen use and hysterectomy, should call for a larger study to clarify these observations, which tend to indicate that exposure to estrogen in middle age increases the risks of Alzheimer's disease in old age, and that even medical progestogens offer some protection against it. Although this study might have been bigger and better, it is far better than the junk studies that have been promoted by the pharmaceutical publicity machine. I have seen or heard roughly 100 mentions of the pro-estrogen and anti-scientific studies, and none mentioning this one. And then we have a couple of studies referenced here. Okay, the next section is called Signals in the Abstract, and this is our last section, and then you'll have to come back for part 2. Okay, when I taught endocrinology, I annoyed my tidy-minded students by urging them to consider the potential hormone-like action of everything in the body, and to think of layers of control ranging from sugar, salt, and carbon dioxide. Through the official hormones, to complex nervous system actions such as expectancy and biorhythms. Certain things that are active in very important processes deserve special attention as signals, but they still have to be understood in context. In this sense, we can think of Ca2+ as a signal substance, so the calcium ion, in its many contexts. It is strongly regulated by the cell's energy charge, magnesium and sodium antagonizing it in certain situations, linoleic acid, linolenic acid, arachidonic acid. Their toxicity is potentially prevented by the amide acids and their glucosinoid derivatives, which behave very differently from the familiar prostaglandins as far as they have been compared, can be drastically reduced by dietary changes. Prostaglandins, prostacyclin, thromboxane. Formation is blocked by aspirin and other anti-inflammatory drugs. Adenosine, sleep-inducing protective effect. Adenosine is structurally very similar to inosine, another natural substance, found in meat, for example, which is a component of inosoplex, an antiviral drug. Or immunostimulant, which has been also found to have an anti-senility effect, with the root being senile, right? So anti-senility effect. Adenosine is a free radical scavenger and protects against calcium and glutamate excitotoxicity. That's from a study. Okay, and there's a quote here. Adenosine, or A1 receptors in rat brain synaptosomes, transductional mechanisms, effects on glutamate release and preservation after metabolic inhibition. All right, moving on. It also appears to protect against the relative hyperventilation that wastes carbon dioxide, and endotoxin can interfere with its protective action. Guanosine in the same group of substances might have some similar properties. Thymidine or cytidine, which are pyrimidine-based, are endogenous analogs of the barbiturates, and like them, they might be regulators of the cytochrome P450 enzymes. Uridine in this group promotes glycogen synthesis and is released from bacteria in the presence of penicillin. And we have iron, regulator of mRNA stability. Heme synthesis reacts with reductants and unsaturated oils to produce free radicals and lipid peroxides. Its absorption is increased by estrogen, hypothyroidism, anemia, or lack of oxygen. Glutamate and aspartate, excitotoxins, and GABA, an inhibitory transmitter. These have metabolic links with each other, with ammonia, and with stress and energy metabolism. Estrogen and acetylcholine, excitotoxins. See Savolainen et al. 1994. The information on this is so overwhelmingly clear, and the publicity to the contrary is a horrifying example of the corruption of the mass media by the drug industry. And we have endorphins, stress-induced, laterally specific, involved in estrogen action, antagonized by naloxone and similar anti-opiate drugs. I have proposed that the endorphins can cause or sustain some of the symptoms of aging. Naloxone appears to be a useful treatment for senility. Again, the root there being senile. We got several studies there. And then we have endotoxin. Anti-mitochondrial action causes elevation of estrogen. It synergizes with unsaturated fats, and naloxone opposes some of its toxic effects. Urea, cholesterol, structural stability of proteins and lipid-protein complexes. Things that act directly on the water structure. I think all of the natural regulators have an effect on the structure of water, but some unusual substances seem to act primarily on the water. Noble gases, for example, have no chemical effects, but they tend to form cages of water molecules around themselves. Camphor, adamantane, and the antiviral drug amantadine probably have a similar water structuring effect, and amantadine, which is widely used as a therapy in Parkinson's disease, has an anti-excitotoxic action. And then next time we'll be reading part 2. Like I said, a lot of information, and then here's all of the references at the bottom, so feel free to read those in your own time. Hopefully you didn't get too lost. I know this is supposed to help people be able to listen and have it in an auditory medium versus visually, because I know some of us get a little busy and don't have time to read these. And that's why I'm doing this. I know I benefit from being able to listen to things, and this is very important to get out there. So that's, again, why I'm doing this. So hopefully I didn't lose you there, and please let me know in the comments if you have any suggestions. Appreciate it, and we'll see you next time.